Today we will discuss about glycan contribution in cancer hallmark, and along with this we will discuss the glycan based therapeutic approaches. Uh, and these things, that is glycan contribution in the cancer hallmarks, is somewhat different from that what we have discussed in our previous videos. Though some of this part might overlap, however it has some additional informations also. So we will start with this sustained proliferative signaling pathways. So in the case of this aberrant glycosylation that has occurred in the cancer tumor cells, what it will do is it will promote this signaling pathway, that is P13K AKT, and because of that it will cause the cells to continuously remain into this proliferative, sustained proliferative signaling, and that helps the cells for this, that helps or contributes the cells towards the uncontrolled growth rate. Next one is there is resistance to cell death. So because of this apparent O-glycanac formations and this lubinek and sarglycin, and this will does is it will form a aberrant glycosylations on the receptors such as FAK, and that will prevent this such as this galactin 3 interactions to this alpha 3 beta 1 integrin that might trigger the apoptotic pathway. However, because this galactin 3 is not able to interact with those integrins, it will not be triggered. It will not trigger the cell death or apoptotic pathway. Next one is this deregulated cellular energetics. So in this case, the reprogramming of this reprogramming within the cancer cells occurs in a way that glycosylations does not require mitochondria. Happened because of this O-glycanations or modifications that might lead to this more O-glycanac formations. And other things are also related to O-glycanac formations, such as this evasion of the growth suppressor that we have discussed in our previous video, that is P53. Because of this glycanac post translation modification of this P53 and this RB E2F1 complex, it will prevent the it will prevent the suppress it will prevent the or it will helps in the evasion of the growth suppressors. The growth suppressors will not functions because of this aberrant post translation modifications. And similarly there is more genome instability and mutations because the recycling of this proteins synthesis pathway, that is protein recycling of this protein through this proteasomal degradations will be affected because of the oglycanations on this ATM that will prevent the proteasomal degradation. And similarly this cymac oglycanations will helps in this uh, replicative immortality, that is it will helps also similar to this resistance to cell death. And this CyMyC also have this aberrant glycosylations in terms of this O-glycanac. And next one is this angiogenesis. Angiogenesis is promoted by different glycans, different aberrant glycosylation profiles that is shown here. One of them is O-glycanac and this alpha-2-cyclic acid, decorin, neoprylin 1,2, HA, and decorin. Decorin already covered. This beta-1,6 glycanac and glycans. So all this will helps in this angiogenesis formation, that is new blood vessels formations, that helps in maintaining the uh, energy requirement for this uncontrolled energy requirement for this tumor cells. Next one is the invasion and metastasis. So in this case the EMT, epithelial to mesomicals, uh, genes are upregulated, and these things has been promoted by this terminal SLE glycan structures that are overexpressed on the cancer tumors cells such as these pancreatic cancer cells. And the other things may also play a role, such as this aberrant glycosylation on the integrins. uh, the let's move forward and Now it is about tumor promoting inflammations. So in this case, the some of these epitopes or the cancer unique epitopes are being detected by the immune response, and that will cause the inflammations, that will trigger the inflammations, and that might even cause problems at a site of the particular site to the of the host body. And some of these glycan structures that cause these tumor promoting inflammations are new 5GCO, Glycnac, SLEAX, and Versi CN. So new 5GCs are is supposed to be that the genes that are responsible for producing new 5GC are being suppressed due to mutations in the human beings. This is current general consensus about new 5GC. Next one is this immune escape. So immune escape are usually done by the glycan structures such as STN, LE, silase, and SD. So what they will does is they will form a uh, barrier or increased glycocalyx thickness in a way that it will act as a physical barrier that will prevent the immune cells to come and interact with the non-self epitopes that are generated on the cancer cells. That is, this glycocalyx prevent the immune cells by making a physical barrier of this aberrant glycosylations profile, and thus prevent the immune cells from interacting with the non-self epitope that are generated on the cancer cells. So now let us quickly cover what are the glycan-based therapies that are being studied, or research are being going on in those areas. First of them, this anti-glycan vaccines, that is aberrant glycosylation that are found only on the cancer cells. Based on those glycan structures, the vaccines are being currently developed so that it can target only the tumor cells but not the other cells. Next one, this glycan targeting with genetically modified antibodies. So in this case, the antibodies are antibodies that will target glycosidase or glycans are been conjugated to these antibodies in such a way that this antibody will only act against this glycan structures and trigger these immunotoxins or trigger the immune response against those glycosidase conjugate. Then and what the other things it will does is it will once it will trigger this immunotoxins, those glycan structures will trigger this ADTC, this antibody dependent cytotoxicity mediated killing. So in this case, the antibodies of the or the immune antibodies are being generated against the non-self glycan epitopes that are being detected in the tumor cells, which is been triggered by this glycan targeting genetically modified antibodies. Next one is this cellular immunotherapy. So in this case, the CAR T cells are being modified with these glycopeptides specific to the tumor cells, so that this CAR T cells can act against the tumor cells that have those glycoepitopes, glycopeptides. Yeah. Next one is this. We can one can easily not easily. One is trying to target in vivo to these dendritic cells by providing them the glycopeptides informations, so that dendritic cells will then use this machinery, immune system machinery, to develop antibodies against those glycopeptides, so that can target the tumor cells and does not affect the healthy cells. So these are some of the ways by which this uh, glycan based therapeutic approaches can be made possible. And we have also discussed how the glycans contributes toward this cancer hallmarks. So that is the motivation behind this glycan based therapeutic approaches. So I hope you got some basic idea or preliminary understanding about the glycans, importance of the glycans uh, towards towards maintaining the healthy cellular functioning within the cells, and any disruption in this glycan cellular functioning might cause mutations that might even lead to the cancer. So if you need to understand more about this topic, that is glycans and this cancer hallmarks, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.